It's currently just finished phase 1. The bottom graph underneath is the sort of the phase 2A in the mice. Now mice obviously aren't covered by Rob's WHO regulations, but you can see that what we want to do is the new drug is the one in pink on the bottom, which says that the mice don't actually see the disease on day 28. All the other drugs on there are drugs which are actually used clinically and work perfectly well. So all this is saying is this is a very tough model. The mice are very tough customers to keep happy, and the drug seems to work. However, the mice usually are a little bit devious, so we're looking forward to seeing whether the human patients actually do the same as the mice. This is to say we have model systems in place to look for drugs which are more likely to hang around in the body, more likely to work, and therefore more likely to be something that's part of a single-dose cure. Manos talked a little bit about the screening, and he showed a wonderful slide where he showed the robotics that they have at Pfizer, where they look through 10 million compounds and say these are not 10 million compounds they found on the street one morning. These are 10 million compounds that they have put together as a result of millions of years of man-years of work. And what's interesting is that all of our pharma partners have these collections, and what's changed over the last 5 years is the ability of the pharma companies to let us do that. And a lot of it has been down to technology. So the fact we can now look at the parasite in a test tube in these high-density formats, so 384s or 1536s, those of you who learn your 96 times table. So immunologists work in 96 wells, and these are just lots of 96 well plates on a plate. So what that means is two things. One is that we can screen much faster, we can screen much cheaper, and it gets to the stage where we're now down to less than $1 per compound that we test. Whereas 5 years ago it was, I'm looking for the STI people, it was $100 or $300 or something like that, and it's much more manual. So you can do things at risk. What we've managed to do, starting with Novartis, Jose in the room did it almost by hand on a 384 well machine, but you can do these screens. We've screened now something like 5 million compounds. Coming out of that come 20,000 submicromolar hits, at which point the numbers people glaze over and they say, this is very interesting. Remember that 5 years ago, we used to get excited when we saw a 1 micromolar hit from one project. So we have LOIs where they say we have one of these. Now we can yawn and we can say, well, we have 20,000 of these. So what else is good about your compound? And knowing that with malaria we need something which is safe, has a long half-life, tastes nice, all the other things that we have to do, then to have the richness of those 20,000 compounds is really important. The other thing is, as Peter mentioned, we have the genome of the parasite. So we know that there are 5,600 and something, I always get the numbers wrong, genes. The clever people in chemoinformatics tell us there are 200, 300, 400 targets. We've actually worked over the last 10 years with all the academics on 90-something targets. So we're working our way through having assays for all of the targets available. So you get this convergence of high-throughput technology and also being able to screen all the genes and knowing what the enzymes in the parasite actually are. The nice thing, if we had, this is Geneva, so normally you have finance guys in the room. Normally everybody says screen to phase 1 within 5 years, genes screen in 5 years. This is usually a little bit of an exaggeration, and here actually we have a couple of cases where it was done. So it is doable, it is possible, but it's only possible because of our partners. About 3/4 of the partnership is with the pharma industry. An important 1/4 is with the good academic centres, and there are some stunningly good academic centres as well. What else are we going to do? Well, we're not going to repeat all of that for all of the other bits of the life cycle of the parasite. So currently what we're doing is we call malaria life cycle fingerprint. So just understanding how the drugs that we have work against each different stage of the life cycle. So against the sexual stages, against the hypnozoites, things like that. And actually what's interesting about malaria is that there is no systematic database of all the interesting drugs in all the interesting stages of the diseases. People have done a little bit of it here and there. What we're able to do, because we tend to work with everybody, is we can assemble their compounds and put them through standardized assays. And interestingly, once you do that, everybody else who has a standardized assay comes in and says, well, can I do it as well? So we can build up quite an interesting database of malaria parasitology just through these assays. I said one of the important things for the future was something against Vivax. Now, Vivax is generally considered to be a relatively benign disease by people who've never had it. The fever, I've never had it, but the fever peaks when you see it are much worse. And of course, it's not curable by just the standard treatment. you have to wipe out the hypnozoites, and that's a 14-day treatment. And as most of you know, if we asked you to take 14 days of antibiotics, then you would miss out the last week, and that generally seems to be what happens. The problem is we have a drug against it, primaquine, which was discovered by the Americans in 1949, I guess. And as Peter showed then, we have this problem that G6PD, which is an enzyme in the red blood cells, actually correlates with malaria because normally it protects you, but it does make the red blood cells more fragile. And here we have an issue that this particular class of drugs are actually not very good if you have a G6PD deficiency. So what we're doing here is we're trying to develop tafenoquine, which was originally developed and discovered by the US military. And we're trying to develop it specifically. It's been into soldiers, but we're trying to develop it for our patients who are women, small children, moving down the cascade, and to try and find out what is actually the safe dose. What this project actually brought out to us is that in Vivax malaria we have a big problem. We have no cellular assay, we have no animal assay, and we have no clinical assay. But apart from that, we're okay. And so just getting that message across was quite important. And so now we're starting to get to the stage, working with the clinical community, that we have a human test situation, which we haven't really had since the Korean War. We have animal assays in monkeys, which we're now trying to simplify down. And we have the first glimpses of activity in cell biology assays. Now, this is not because it's particularly complicated. It's just the resource hasn't been there, the finances haven't been there, and so nobody's encouraged to go into it. Partnerships. I think, as I said before, I always thank everybody, and I miss 2 or 3 people out. So for the 2 or 3 people, I think we've got the MIP consortium missing from here and the IPTI consortium, which is bad because Pedro's here. But say, in the future, then we have phase 4, we have lots of work. We are not the agents of phase 4, so phase 4 is out in the community. We're dependent on all those people up there to do the work, and we can help liaise with our pharma partners, we can help oil the wheels, but that's pretty much what we're doing. With our pharma partners, we're working all the way from screening to phase 4. We have some amazing partners out there. Without them, nothing would really be as easy as it is. In fact, you can almost say nothing would be possible if you add in the academic partners as well. I am privileged. I've worked with several scientific advisory boards. It's fair to say that, so we are privileged to have some of the finest minds in the world advising us and actually telling us what they think, which is spectacular. We have a global safety board that we set up this year where we were privileged that physicians who have much better things to do came in and advised us on the safety of medicines, which is really important to us. We're privileged with the Gates Foundation, with the Wellcome Trust, that we often put them down as sponsors, which makes them sound like guys with big sums of money, and they are guys with big sums of money, but they also bring vision and direction as well. And finally, just to say, none of this, I mean, we do this for the patients, for the children, for the people who have the disease, but of course nothing's possible without the clinical trial sites, and nothing is possible without our patients and the consent of their families. And we should always be grateful for their participation. So my conclusions, we have a strong portfolio of new medicines all the way from discovery to delivery, and George is going to come and tell us all about the delivery aspects. In the last 5 years, there's been a major sea change. So the technology, the screening technology, the genome sequence, and so on really does mean that we have a lot more tools. And that what it's meant as well is that our mini portfolios, the collaborations we have with our pharma partners, are now well supplied, moving forward. We have new development candidates coming in. We like to track, just how many new phase 1s we do every year. And actually, we're taking 2 or 3 molecules every year into man for the first time. So that's a good measure of success. And they're being pre-selected. Now we can look at them and say, not just do they deal with the red blood cell stages, but what are they going to do in eradication, and are they really the best things to develop? And finally, just to finish where I started, which is to say, this is all about you. This is all about our partners. This is all about the work that you've done to help us here. And that's the key to our success. And when you look at that pipeline and you look around the room, you can associate virtually everybody in this room with 2 or 3 of those boxes. I think that's pretty impressive for an organization. And I think I'm still allowed to say that as the newcomer after 2 years. But it's been a great journey. So thank you.